So we're having our test, lab test 3 on Thursday at regular lab time. So we'll go ahead and get started. Page 121 in our lab manual is Laboratory 10 on human genetics. So all these questions are based on humans, just to be clear, since the title of the lab is Human Genetics. And I want to remind you guys that humans have 23 pairs of chromosomes. And if you don't remember what that looks like, just glance at page 123. Page 123 is a human karyotype. So that's what we're referring to as far as the chromosomes of a human. There's a human karyotype right there. And the first question is, where are autosomal characteristics carried? And I'm asking specifically, what chromosomes do we consider autosomal? Does anybody know the answer to that? What chromosomes? Chromosome 1, any others?
1 through 22.
That's excellent. Thank you, Tim. It is 1 through 22 are the autosomes. 1 through 22 are the autosomes. Can you guys see that okay? All right, super. And then where are the sex-linked traits carried? The 23rd pair. That's good. And that's, you can tell there, that's the X and the Y. And I'm just glancing at page 123. Looks like we have a little girl right there. No, that's a boy. Hello. Everybody's okay on number 1? Okay, what chromosomal abnormality is associated with Down syndrome?
Chromosome 21.
Is it 21? And how many of 21? 3. So yes, you're right. So Katie said that trisomy 21. So that means they have 3 21st chromosomes. And of course, a normal karyotype would just have 2. Now, trisomy 21, if you want to read about it, that's on page 122. Make sure that you can look at a karyotype and be able to tell that it is Down syndrome.
Thank you.
Mm-hmm. There. Okay, number 3. How can you tell the sex of an individual in a karyotype? Where do you look? 23rd pair. So you're going to look at the 23rd pair. What if you see 2 large chromosomes at the 23rd pair? It's a girl. How about if you see 1 large, 1 small? It's a boy. That's good. Okay, what chromosomal abnormalities are associated? And then I made this list here. Now the list, guys, also is on page 125. Let's glance at page 125. That's where I got this list from. This may seem familiar to you. The Turner syndrome, Poly X, Klinefelter syndrome, and Jacobs. So let's start with Turner's. What chromosomal abnormalities associated with Turner's? Anybody?
X.
Only 1 X. So we show that by putting the X and then what?
0.
A 0. So instead of a quote, normal female being XX, a Turner's female is X and then a 0. Okay, on Turner's. All right, how about Poly X? An extra X. So we're going to write 3 X's for this one. And guys, again, I think you did this on page 125, table 10.1. Klinefelter's. XXY, that's good, Madison. XXY. That would be a male. That would be a male. That's good.
So anytime there's any form of Y, it's a male?
That's true.
Would it look like a female?
No, they tend to be kind of tall and lanky, maybe some breast development. Let me tell you why this Klinefelter's isn't a huge problem for a male. It's because you guys may remember one of the X chromosomes in every cell is turned off. It's coated with proteins. That's true for every female in the room. In every one of your cells, only 1 X chromosome is active, information. No, it's pretty much like a boy. It's got a little bit of extra DNA, but it's mostly turned off, so that's why it's not a huge deal. Good question. Okay, and guys, to finish up, what about Jacob? XYY, very good. And then, again, there's no problem here because the Y chromosome is so very small. think it's got like 7 genes on it. It's very small. All right, lastly, on page 125, make sure you go back and study those karyotypes that were in the middle of page 125 and that you could figure out which disorder goes with which karyotype. Because that's the most important thing. As far as just descriptions of the disorder, you've been tested on that on lecture tests, but now visually seeing the disorder is what I want to test you on in lab. So spend time looking at those karyotypes on page 125. Okay, I have if a person is homozygous dominant, like big A, or if they're heterozygous, like big A, little a, what characteristic will be shown in their phenotype? Will they look dominant or will they look recessive? Dominant. Okay, good. Dominant. That's an easy question. How about if an individual is homozygous recessive, like little a, what trait will show in their phenotype? Recessive. Very good.
Will we have to know those recessive traits?
No, I always tell you what's dominant, what's recessive. Number 6, you would expect the dominant trait to be the most common trait in a population. Is this always true? No, it's not. How come? Okay, let me give you an example. Huntington's is that disease where you die from nervous degeneration. Your nervous system goes downhill when you're about in your 40s. That's dominant. But that's not very common in our population. Can you figure out why that trait's not common in our population? They can reproduce, but it's just not very common in our population. Can you think of why? Is it favorable to have Huntington's? No. And so just because a trait's dominant doesn't mean it's favorable. So natural selection, you guys heard of natural selection? Of course you have. Natural selection doesn't always favor the dominant trait. Does that make sense? Okay, so why? There's lots of reasons why, but that the dominant trait may not be favored. I just have to share this with you real fast. You guys know that if you're looking at humans, if you go towards the equator, most humans that are living in their native lands have darker skin, darker hair, darker eyes. Everybody shake your head. And if you go away from the equator towards the poles, most native humans have lighter skin, lighter eyes, lighter hair. Now, isn't that recessive to have light skin, light hair, light eyes? Yes. So there must be a reason why that was favored. And why humans that live at northern latitudes are so fair skin toned, hair and eyes are so light. So people are trying to figure out why. And it's because of vitamin D. It's because people with dark skin have a lot of UV protection, and you need that, lots of UV protection if you live on the equator. But do you need UV protection if you live up in Alaska? No, you don't need so much UV protection. However, there's not as much sunlight out there in Alaska or Norway or, I don't know, the Soviet Union. There's not so much sunlight. Do you guys know vitamin D? You have to get sunlight to make vitamin D. Have you heard about that? You have to expose yourself to some sunlight. Well, if you're living on the equator, you're getting plenty of sunlight. So you're making vitamin D no problem. But if you had dark skin and you lived at these higher latitudes, the dark skin wouldn't let much of the sun penetrate your skin, so you wouldn't be able to make vitamin D as well. So dark skin is actually not favored due to natural selection closer to the poles, whereas dark skin is favored near the equator. And it's because of the sun and making vitamin D. That's just an example. Okay, I think this is what Tim was talking about. Do we have to memorize what's dominant, what's recessive? And of course the answer is no. So here I say it in number 7. If I give you information about which trait is dominant and which trait is recessive, be able to work the genetics problem. So what am I talking about specifically? Page 127. Specifically, question number 1. Make sure you go back and look and read about how Nancy and her family there have attached earlobes, and we worked out some genetics there. And then page 128, Number 2 about Joe, and number 3 about Henry. And then on page 128, we also worked out questions on cystic fibrosis and Huntington's. So that's specifically what I'm talking about. So I'm just going to write down that you need to make sure and look at page 127 and 128 and go back and reread those genetics problems and make sure you can work them. But once again, I'll tell you what's dominant, what's recessive. Okay, so just for fun, I thought we would do this. 2 people, this is not on your review sheet. 2 people with widow's peaks, and widow's peak is dominant, have 3 children. 2 of their children have widow's peaks, and 1 of their children have a straight hairline. What are the genotypes for each? Because everybody look at the problem. I got 2 people that are mating, and they both have widow's peaks. Then they have 3 children. 2 have widow's peaks, 1 does not. So I wrote down a key up here at the top that the widow's peak is dominant and having a straight hairline is recessive. So I'll give that to you. You don't have to memorize that. Okay, so we have to figure out what are the genotypes of the parents. That's good, Stephanie. Stephanie, do you have a review sheet? Good. How come they couldn't possibly be big W, big W? Because of one of their kid is recessive. That's good. Okay, now what about the children? 2 of them have widow's peaks. What about the 2 children with widow's peaks? Are they big W, little w? We don't know. They're either big W, little w, or what else? Yeah, so what would we do?
Big W.
That's good, Tim. So the 2 kids that have widow's peaks definitely are big W, but we don't know if they're big W or big W little w. So we know big W question mark. Okay, but what about that 3rd kid? 2 little w's. We know that one for sure. That's you may remember in a few minutes we're going to do some Hardy-Weinberg. You may remember that when we do Hardy-Weinberg, we always start with this one. This one right here. Q squared is that one right there. And the reason is that's the one we know for sure, their genotype. The dominants you don't know for sure, but the recessives you do. That's why we always start there. Okay. This is question 8, true or false? Sex chromosomes carry only genes that relate to the sexual characteristics or sexual functions of an individual. Is that true or false? Anybody? The X chromosomes only carrying genes about sex, the Y chromosomes only carrying genes about sex. Is that true or false? Nope, it's false. Now we need an example of a gene on one of those chromosomes that has nothing to do with your sexuality. You may know a gene on one of those chromosomes that has nothing to do with you being a boy and, you know, having all your boy male parts and so forth, or nothing to do with being a girl and having all your girl male parts. Girl male parts. All right, so I'll give you an example. Color blindness. Being able to see in color. Does that have anything to do with your sexuality? No. Has nothing to do with your sexuality. Color vision is on the X chromosome, but that has nothing to do with your sexuality. I'll give you just another one. Blood clotting, being able for your blood to clot when you cut yourself, is on the X chromosome. Has nothing to do with your sexuality. It has to do with whether you can clot your blood or not. Okay? So that's just a couple of examples of genes that are on the X chromosome, have nothing to do with your sexuality. Pretty okay on number 8? Okay. Number 9. How are X-linked traits? That means traits that are only on the X chromosome. These are traits that are carried on the X chromosome. How are X-linked traits inherited differently in males and females? Okay, so who gets 2 X chromosomes, males or females? I'm sorry? I didn't hear you. Females. Okay, so females, which this is the symbol for female, we get 2 X chromosomes. This is the symbol for males. What do they get? An X and a Y. Okay, now I'm saying that X-linked traits are only on X chromosomes, so we put them as little superscripts up here. Everybody look how a female gets 2 chances to inherit every trait on the X chromosome, but boys only get 1 chance. Okay? So X-linked disorders like being colorblind or hemophilia where your blood can't clot, who would that show up more often in, boys or girls?
Girls.
It show up more often in girls? Nope. You know anybody who's colorblind? They're all guys. Yeah. You probably don't know anybody who has hemophilia, but colorblind is pretty common in our population, and they're all guys. Okay, now let's just pretend here that big C is normal. I'm writing this down. Big C is normal vision, and little c is colorblind. Now you look at why colorblindness shows up more often in males. Everybody look at it. Why would colorblindness show up more often in males? How many chances does a girl get to inherit information for vision? Twice. But boys? Once. Okay, so look. What if you had a girl was big C, little c? See, she got 2 chances to inherit the colorblind gene. She inherited normal vision and the colorblind gene. Does she see in color or does she not? She sees in color. Okay, but here, if the male gets a big C, he sees normal, but if he gets a little c, he's colorblind. We only got 1 chance to see in color. Girls get 2 chances, right? So these things show up more often in males than females. So make sure that's in your notes, that X-linked traits show up more often in males than females. However, where do little boys get their X chromosomes from? Their mom or their dad? They get their X chromosomes from their mom. What do they get from their dad? The Y. Okay, so where do these genes kind of hide generation to generation? In males? Now you can't hide it in males. They're hiding in females. Okay, could you look at this lady right here, talk to her, look at her, talk with her, and tell that she's carrying the colorblind gene? No. Could you talk to this male and figure out that he has the colorblind gene? Yeah, you just say, hey, are you colorblind? Or give him the colorblind test, and he'd say, I can't do that. Okay? So I would say this in my notes, that females tend to carry the trait, but males show it. Females tend to be the carriers. It's like they're hiding it. They don't know they even have it. They don't even know. And then they pass it to their boys, but they don't even know that they have it. Hey, good morning, Karen. You need a review sheet? Okay, so I have, how are X-linked traits inherited differently in males and females? Males tend to show the trait. Females tend to carry the trait. But okay, on number 9. Number 9. Okay, great. Okay, I have no pedigree symbols. Okay, so what is a circle on a pedigree? A circle. That's female. Good. A circle is female. And what is a square? Male. Do you guys know these symbols for male and female? Yes? I think this is the symbol for Venus. The female symbol is the symbol for Venus. The male symbol is the symbol for Mars. Venus and Mars. I'm just telling you what they are. Okay. All right, does a pedigree show genotypes or phenotypes? So let me just show you a pedigree. I made up one here. Is this telling me a person's genotype or a person's phenotype? What they look like or their actual genes? It shows me their actual genes? No, it's just telling me what they look like, whether they're normal or not. It's not telling me if they're big W, little w, or little w, little w. I've got to figure that out by myself. If I just glance at this pedigree, what I know is, are you normal or do you have the disorder? How do you know if the person's normal? If it's not colored in. How do you know if the person has the disorder? If it is colored in. Everybody okay with that? Okay, that's just your basic stuff. Okay, now if you link a circle and a square together like I have done right here, what does that mean? Nope, they're not related. I sure hope they're not.
Oh, they're couple.
They're a couple. They are a couple. Very good. I don't want them to be related.
I thought you did the ones on the bottom.
No, this one right here.
Together.
Yeah, right here. So those a mating pair. Are you okay with that? We don't want to know if they're related. We just want to know if they're a mating pair. Okay, then you drop a line down, right? And then what are these? Those are their kids. Everybody's okay with that? All right, good. Okay, so on the previous screen, I've said predict how a trait is inherited. Everybody, you have to know this. Page 131. What am I talking about? I'm talking about the top of page 131, table 10.3, inheritance pattern. There's 3 choices there. So this is page 131, table 10.3. And I'm talking about autosomal dominant, autosomal recessive, or X-linked recessive. Those are going to be your 3 choices on the test Thursday for inheritance patterns. So I need you to study this table, especially the clues. The clues. That's page 131, table 10.3. Okay, so if you'll glance at those 3 choices, let's look back at my pedigree that I came up with here. See if we can figure out, is this one autosomal dominant, autosomal recessive, or X-linked recessive? Autosomal dominant, autosomal recessive, or X-linked recessive? What do you want to go with? I hear a nomination.
I said it was recessive, but I don't know which one.
Okay, so Katie's nominating recessive, and I can see why you'd pick that because, look, neither parent has it, but one of their 3 kids does. Okay, and then that kid married somebody else, and one of their kid doesn't have it, one of their kids do. Check this out. Neither one of these parents have it, and again, one of their kids do. So I like that recessive idea. Now we've got to narrow it down. Is it autosomal recessive or X-linked recessive? Okay, Alaska's nominating autosomal. Tell me why, Alaska. And I agree with you, Alaska.
Well, it says it's only carried on the X chromosome, and then it was saying that it's usually found in males, and it's often passed from like grandfather to grandson. So it's skipping a generation, and it doesn't do that.
It doesn't do that. The people who have it up here, we got 1 male and 2 females. So that really doesn't fit the X-linked recessive pattern. If you were looking for X-linked recessive, you would be looking for mostly males having it. And then, like Alaska said, skipping generations. But it didn't skip a generation here, and of the people who have it, it's only 1 male and 2 females. So it doesn't seem to matter if you're male or female on this one. So let's go autosomal recessive. So I'm going to write that down up here at the top. Autosomal recessive. Okay, now we're going to use the letter A to stand for the trait. And remember, this is an autosomal recessive disorder. And we're going to see if we can figure out some of the genotypes of these parents and their offspring. So let's start at number 1. Everybody look at number 1 up here on the screen, because this one, I just made this one up. Okay, generation number 1, the mother, which is generation number 1, individual number 1. Using the letter A, what is her genotype? Ready? Katie Lightfoot said big A, little a, and I agree. She doesn't have the disorder, but she's obviously carrying it. How come it's obvious she's carrying it? Because of this sign right here, correct? Okay, what about the husband? Big A, little a. He's the same thing, correct? Okay, I like generation 2, individual 2. Right, check out their firstborn daughter. Generation 2, individual 2. She doesn't have it. What's her genotype? She's big A, little a also. How can you tell? Because of her daughter. Let's talk about her daughter. Generation 3, individual 3. What's her genotype? Little a, little a. That's good. Okay, I got one more I want you to do. Do generation 3, individual 4. Generation 3, individual 4. Big A, little a. How do you know she's big A? She doesn't have it. How do you know she's little a? Because her dad did. So she's got to be big A, little a. Okay, this is the kind of thing I need you to do on the test Thursday. Job number 1, figure out the inheritance pattern. And you have 3 choices: autosomal dominant, autosomal recessive, X-linked recessive. You've got to do that first. And then figure out people's genotypes. But are you okay with that? Yes? Okay. Susan has blood type A. This is number 11. Susan has blood type A. Jack has blood type AB. What are the possible blood types of their children? Well, I wanted to remind you about blood types. I made this chart. You could find this chart in your lecture notes. If you want to write it down, feel free to do so, because we haven't gone over it in quite some time. So when I say a person's blood type A, I am referencing their phenotype. Blood type A is a phenotype. Blood type B is a phenotype. Right, okay with that? And then here's their genotypes over here. Now, can you look at these genotypes and tell me, is A dominant or recessive? It's dominant. Is B dominant or recessive? It's also dominant. Is O dominant or recessive? Recessive. Everybody see that okay from our chart? Feeling comfortable with that? Okay, now, I think it said that Susan's blood type A. Let's go back to that chart. Should we assume that Susan is AA or AO? For sure. You assume Susan's AO because you don't know. You can't tell, so you assume that she's hiding the O allele. Just assume that. It's okay with that. All right, so we're going to go ahead, and I've got this problem here. You agreed with me that we're going to assume that Susan is AO, so I went ahead and put the O in there. Let's look and see how this would be solved. Hey, good morning, Alexander. Do you need a review sheet?
Yes, I do.
Okay, you want to come on up? Missed you last week. Okay, so here is Susan. I put Susan here. And here was, what was his name, Jack? Here's Jack. Okay, now could Susan and Jack have a baby blood type A? Yes. Could Susan and Jack have a baby blood type B? Yes. Could Susan and Jack have a baby blood type AB? Yes. And could Susan and Jack have a baby blood type O? No way. There's no way. Let me see that. Okay, so if I gave you this problem on the Scantron, you would have to color in blood type A, blood type B, and blood type AB. Would you color in blood type O? No, it's going to be one of those choose all that apply. You know those questions? It means it has multiple answers. Is everybody okay on this blood type question? Okay, does anybody need this chart? Yes, ma'am.
Did you say whenever it's blood type A, it's the phenotype?
That's a phenotype. And then, Stephanie, we said if I give you that they're blood type A, you're going to assume their genotype is AO. What if the problem told you you had a person who's blood type B? What should you assume their genotype is? BO. Okay, if I tell you somebody's blood type AB, then what is their genotype? AB. What about blood type O? OO. Everybody okay with that? Okay, great. Oh, I'm sorry. So we're going to go now to exercise 11 on DNA, biology, and technology. So I've got these words up here, replication, transcription, translation, which you guys can look up in your notes, so no problem there. But I wrote this down. I remember putting this on the board downstairs before because it's one of the coolest things in biology. I have red arrows up here, and I think each of these red arrows go with one of those words. So I want to see if you can help me figure out which arrow goes with each word. Tim?
I have a question about the previous question.
Okay. The one about the blood type?
On my, oh, never mind. Nothing on my page.
Okay. Okay, so everybody's okay on number 1 about replication, transcription, translation. These are our choices. Okay. So help me fill this out. I got this first arrow right here that shows DNA can be copied to make more DNA. What's that called? Replication. So we're going to put replication here. Okay, my next arrow says that I can make RNA from DNA. What's that one called? Translation? Is that true? That's transcription. I did. That one's transcription. Can make RNA from DNA. That's transcription. Okay, how about making an amino acid sequence or a protein from RNA? That's translation. You guys can figure things out fast. Now, if you want actual definitions, feel free to look those up. You've got them in your lecture notes. Number 2, what are the 3 parts of a DNA nucleotide? Well, I'm going to draw one, and then we'll talk about what the 3 parts are. Okay, let's talk about the P with the circle around it. What is that? Phosphate. Nice job. Phosphate. How about the S? It looks like it's living in a house. That's the sugar. And let's see, this is DNA. So what's the name of that sugar? Deoxyribose. Sugar will not be a choice on the test. That word is not even on the test. The word sugar is not on the test. But deoxyribose is. Ribosome. Hello. Ribose. I can spell. That's better. Okay, B with a box around it. What does that stand for? Base. And if we're doing DNA, what are our choices for the base? DNA. What bases are in DNA? A, T, C, G. That's good. Everybody okay on number 3?
Are you going to give us something like this? Or is this the
Could be a choose all that apply. Choose all that apply. Yeah. So don't pick uracil. Yeah. And don't pick ribose. Okay? Okay, the second part of number 2. In DNA, what parts make up the latter sides of the DNA molecule? Okay, here's our parts. Phosphate, sugar, and base. Which of those make up the backbone? The sugar and the phosphate. That's good. The sugar and the phosphate. That's what makes up the sides of a DNA ladder, so to speak. And what make up the steps of the ladder or the rungs? Bases. Let's talk about those. A. Who does A always go with? T. And then what does G go with? C. How come I put 2 lines between A and T, but I put 3 lines between G and C? Because how many hydrogen bonds there are? A and T just have 2 hydrogen bonds. C and G have 3. Now, just as a side note, guys, there's some excellent artwork on that you might want to mention. Page 136. You want to just glance at some artwork when you're studying. Page 136 in your lab manual is pretty good. Okay, so the actual test, we'll have the models that we built. So I took a picture of our models that we built. I told you guys to do the same, take pictures of your models. And so I drew some white lines to some of the things that are here. So let's just start with this top white line right here. Can you tell me what this white tube stands for? Phosphate. That's good. How about this black pentagon, the second white line? It's going to that black pentagon. What does that stand for? Deoxyribose. That's good. Now, I could have drawn this third white line to any of these colored tubes. I just drew it to this blue tube. What does that stand for? The base. That's good. And then what about this solid rod that's in the middle holding the bases together? Hydrogen bond. That's good. Okay, what's this whole molecule called? What's on the screen right now? What is that called? DNA. Okay, don't miss the easy stuff. Okay, that'd be sad if you could do all the parts, but you didn't know it was DNA. Okay, that would be kind of sad, right? Everybody's okay on that one? Okay, super. Okay, number 3. Predict the base sequence for a complementary strand of DNA. First of all, how do you know this is DNA? Glance up here. I just made up a sequence. Doesn't have U, instead it has T, okay? Now, let's go ahead and practice this together. If you don't mind, glance up at the screen and help me out. So here's a 3'. So what would I write down here? 5. Okay, and go ahead. A. So that's something you can easily do? Yes? So I'll ask you to do that. Give the complementary piece that would go with a single strand of DNA. You also practice that in your lab manual, the bottom of page 138. We did that as a class if you want more practice. That was number 3. Okay, number 4, what are the differences in structure and differences in composition between DNA and RNA? Okay, I'm going to get us started because you already told me one. You said that DNA has T, but RNA doesn't. Instead, what does RNA have? U. Good. Go ahead and put the word stranded in there. Okay, I'm just going to draw lines so we can see these are things. And there's one more difference we can think of. Good. Alaska said DNA uses deoxyribose, and what does RNA use? Just ribose. And then Alexander added that DNA is double-stranded and RNA is single-stranded. Everybody see that okay? I just made a little table. We're all good? Okay. Okay, so I took another picture. First of all, what's the name of this molecule that I took a picture of? What kind of RNA? Ooh. M, that's right. This one's our mRNA. I'm glad we had this chat. Okay, now RNA is also made out of sugar, phosphate, and bases. But it's single-stranded, like Alexander said, not double-stranded. So let's go through and see if we can label the parts. Let's start with this tube right here. I put the pointer on the red tube, but it could have been the black or the blue tube. Any one of these colored tubes. What would that stand for? The base. That's good. How about the white tube down here at the bottom? Phosphate. And how about this blue pentagon? That's ribose, the sugar. That's good. Everybody okay with that one? Okay, number 5, what's a codon? I'll get us started. is 3 bases of mRNA. It's 3 bases of mRNA. Now, what do these 3 bases code for? I'm sorry? Excellent, Katie. Amino acid. Katie Lightfoot's correct. Codes for an amino acid. You guys remember this? And it's every codon with the amino acid it codes for right here. Is okay with that? Okay, and then I have what organelle is associated with the process of translation? And I'll give you a hint, it starts with an R. Thank you. Is that Tim? Good job. Ribosome. The organelle that helps make proteins in the cell involved in the process of translation is the ribosome. Okay, now this next one for number 6, let me see. Number 6 says if given a DNA code, be able to transcribe it to messenger RNA. So I went ahead and put a code here for us to practice to see what happens. What does this word transcribe mean? What are we supposed to do? Make RNA. Okay, so this says a 3, so what would be right here? 5. Go ahead. A, U, okay. Now that's doing transcription. That's all I'm going to tell you to do is transcribe this. You have to know what the word transcribe means. It means to make RNA. And we practiced this at the bottom of page 141. If you want to look at another example where you practice doing that, it's page 141. Okay, I took another picture. What process am I taking a picture of here? Not sure? Translation. Karen's right. This is translation. Okay, we have to label all the parts.
How do you know that's translation?
Let's talk about it so that if we can label all the parts, I think we can figure out how we know it's translation. So let's start with this top thing to have a bracket on. I am referencing this whole long molecule here. What is this whole long molecule up here at the top? mRNA. Good. So you may see any DNA in this picture. Well, DNA is not involved in this process. No DNA is involved. Okay, now what about this blue thing that kind of looks like a hamburger bun? That's the ribosome. Okay, and then how about, I'm just going to put the circle around it. How about this thing right here? I've got 3 of them if you'll notice. I've got one right here, one right here, one in the middle. What is that? There's an amino acid. That's the black part right there. What's the whole thing? It's tRNA. Who told me that? Alexander's correct. This is tRNA. So going back to Tim's question, how do you know that's translation? Well, what are we doing? Pretend. What are we doing here? We're making an amino acid sequence because you guys just told me here's an amino acid, here's an amino acid, here's an amino acid, and we're putting them in order. So what was that process called where you make an amino acid sequence from RNA? That's translation. And then just to back up for just a second, I have this question. What organelle is associated with translation? I think Tim's the one who told us the answer. He said the ribosome. So if you see the ribosome on the tabletop, you're doing translation. Okay, ribosomes are only involved in translation. So those are all clues. I'm just trying to give you some clues how you would know that was translation. Guys, what if I had a DNA molecule pulled apart and I was making an RNA molecule from it, what would I be doing? Would I be doing translation? That's transcription. Yeah. So you really do need to know that little sentence that we have that has DNA with the circle around, that's replication. When you're making RNA from DNA, that's transcription. When you're making a protein from an RNA molecule, that's translation. You actually do have to know that. Okay, first of all, what is this whole picture a picture of? This whole thing. tRNA. Ready? Okay, this is tRNA. Shows very well. Okay, what do you call these three colored tubes? They're bases of what? I'll give you choices because the test is choices. Codon or anticodon? This is an anticodon. Okay, and then what is this black thing at the bottom? Amino acid. Very good. Okay, so the whole thing is tRNA. I'm going to write it again because it didn't show up very well up there. This whole picture is tRNA. At the top you have the bases of the anticodon. At the bottom you have the amino acid. And the reason I put this picture on here is because I think it goes with number 7. This picture goes with question number 7. What components are necessary for translation, and they're carried by the tRNA molecule? I need 2 things on the tRNA that help do translation. The anticodon is one, and the amino acid is the other. It's an A. But okay on that one. Okay, and then I'll help you with this. What is an anticodon? I'll get us started. It is 3 bases. So let's go back and look. There they are. There's the 3 bases. Oh, it is 3 bases. But there's specifically 3 bases complementary to a codon. They're complementary to a codon. They are 3 bases, but they're complementary to a codon. So we're going to practice that. We're going to practice that. And we have practiced it before. I just want to point out where you have done it before. On the top of page 143, you practiced doing anticodons at the top of page 143, table 11.7. So let's practice some more. Let's practice some more. So I have some codons here. How do you know these are codons? Well, I told you they're codons, but how do you know that's RNA? Because uracil's there. And then I'm asking you to give me the anticodon. So go ahead and go forward. Here's the first codon, CGC. What would be the anticodon? G, C, G. Okay, and go ahead and give me the second anticodon. A, U, A. Everybody okay on making anticodons? See, it's 3 bases. Anticodons are 3 bases, and they're complementary to a codon. Okay, so we have it in words, then we can practice it and see that that's what it is. Okay. All right. I ask you to translate this. This is not on our review sheet. I want to ask you to translate this. So what am I asking you to do? Do the amino acids. So we're going to have to find this sheet. Let's look in our lab book or wherever you keep your stuff. I have no idea. And see if you can find your genetic code. Okay, glance through your lab manual. Maybe you shoved it in there. If you have your genetic code. Genetic code looks like this. And don't forget, the genetic code will always be given to you. The genetic code will always be given to you. If you don't have one, come on up. Sir?
we gonna have to have our copy of that for the test?
No.
Or is that just-
It'll be, it's, remember how on the lecture test I scan it into the doc? It's the same thing. And so here, what I would do is this would be at the bottom, and then there would be a question at the top, so you could just glance down and see this. Because there would be no reason to memorize this, and yes, I'll always give you this. Okay? Okay, so let's go ahead and get started. Then everybody has one now? Is there anybody who needs one? And everyone, let us use 3-letter abbreviations for our amino acids. The first 3 letters is what I'll be doing on the test. I'll use the first 3 letters for abbreviations. Okay, here we go then. What did you get for AUG? I didn't hear anything. What'd you get? Okay, MET. That's good. Okay, what'd you get for CGC? Arginine, okay. What'd you get for UAU? TYR, tyrosine? Is that right? What'd you get for UAG? Stop. Everybody could say that one loud enough. So you're feeling okay with this? All right. All right. Since you were digging around your lab manual, I want you to dig around and find another piece of paper in there. It was called electrophoresis. So I'll look around for our piece of paper in our lab book called electrophoresis. Electrophoresis. Dig around, see if you can find that one. Electrophoresis. If you were here that day, on the back, you took some notes with a drawing of a test tube. So this is on the back of that sheet. If you weren't here that day or you can't find it, then you don't have it. Sorry. Hopefully somebody's going to be nice to you. Okay? It's on the back of that sheet. And so on the back of that sheet, we drew a test tube and we did a DNA isolation using banana. And I have, during the DNA isolation, what role did each play? And the first one was the cell extract. The cell extract. Does anybody know what role the banana cell extract played in this procedure? Source of DNA. Very good. That's where we got our DNA to separate from. Source of DNA. How about the SDS? It's the detergent. What did the detergent do? Mm-hmm. And what did the ethanol do? Anybody? It pulled the DNA. That's good. It pulled the DNA. Mm-hmm. So it actually did the separation. It's kind of like it's a DNA magnet. The DNA is attracted to the ethanol. So the DNA rose up out of the mixture into the ethanol, and then we could see it there. Is everybody okay on number 9, the DNA isolation? Okay, great. All right, so flip that piece of paper back over to where it says electrophoresis. It says, what is gel electrophoresis used for? And I found the answer in the first sentence at the top. Gel electrophoresis separates molecules. You're right. Upon what basis does it separate molecules? Answer still in the first sentence.
Charge and size.
Charge and size. Good job. Okay, now you have to use your memory. Do you remember this day where we used the little gels in the equipment? What made the molecules actually move? Mm-mm. The electricity. That's good. If you don't plug it in, it didn't work. I think somebody almost forgot to do that. Yeah, but we made it. So we all did good. That's good. All right, number 11. If given a protein's isoelectric point, and everybody check it out, isoelectric point is the pH at which the protein has no charge at all. So if I give that to you, and then I give you the pH of the buffer, determine if the protein will be positive or negative. Now, guys, we did that together at the bottom of this page. You see the table that says sample, and it's got cytochrome C, myoglobin, hemoglobin, serum albumin. And then I give you the isoelectric point, and I give you the pH. Does everybody see what I'm talking about? And then we predicted whether it was going to be positive or negative. Okay, so I need you to go back and review that. That's what I'm telling you, to go back and review that. Now, everybody check this out. I did a pretend gel. And these all had color to them. And I numbered them 1 through 5. And then at the bottom, I told you the pH is 7. pH of 7. And I got a couple questions for you. If I gave you this gel, here would be the questions I would ask of you. Which of these pigments are positively charged? 2, 3, 4 is not correct. It's 1 and 5. How come it's 1 and 5? Because if you're positively charged, which way do you go? Negative. You guys jump too fast. Before you jump, what do? Look and think. Do I really want to do this? Yeah, you need to look first. That's right, Alexander. Just don't jump. Very good. Okay, so the question was, which of these pigments are positively charged? And everybody agree it's 1 and 5. And why is it 1 and 5? Because they go the opposite direction. Everybody okay with that? Yes? Okay. Which of these pigments, you get to look at all 5 of them, which of these pigments has an isoelectric point closest to pH 7? It's number 4. How come? Because it didn't move very far at all. Ready, check it out. Number 4 didn't move very far at all. If you have a pH close to, I said that incorrectly. If you have an isoelectric point close to the pH, you don't have much charge, so you won't move very far at all. Okay, so I want to repeat the 2 questions that I might ask you of this gel. First, I'd ask you, like, who's positively charged? Or maybe I would ask you, who's negatively charged? Could you figure that out? Yes, everybody's okay on that? And then I would ask you, who's got an isoelectric point closest to 7? And everybody said 4. Everybody's okay on that one? That's good. All right, so that should take us to exercise 12, and we get to flip to the back. Evidences of evolution. Okay, what is evolution? Well, you can find the definition in your lecture notes. I'm going to go ahead and write it down because it's so short. It's the genetic change in a population I'm abbreviating population over time. So we're looking for changes in the genetics of a population. That's evolution. If we see changes in the genetics of a population, the population's evolving. So it's the genetic change in a population of organisms over time. You could find that definition in your notes. But okay, on the definition of evolution. Okay, I also have, how does evolution explain the unity and diversity of life? Okay, the unity of life. How evolution explains the unity of life? Well, all semester I've been telling you that there is a lot of unity in life, like that all cells have DNA, all cells have all 3 kinds of RNA, all cells use the same genetic code, all cells use the same amino acids, all of those things. So evolution helps us understand that because all life has common descent. What does that mean?
We got a common ancestor.
We all have a common ancestor. That's why chemically we're all so similar to each other. Tim?
It's still number one.
This is still part of number 1. Yes, sir, it is. Okay, but I can look around and tell that a mockingbird is different than a human, or that a worm is different than a mushroom. So how does evolution help me understand the diversity of life? If we all have a common ancestor, then why are all life forms so different? What's happened over time? Has life stayed the same over time? No, it's changed, but why? What's been driving the change? Evolution, but what mechanism of evolution? Mostly natural selection. Yeah. Okay, so the diversity of life is due to changes due to different environments. Okay, a white rabbit isn't going to make it very long in Texas. Is there a place where a white rabbit's going to make it? Sure, wherever there's snow. Okay, so there's different environments, so nature has selected different traits that are favorable in those environments. Is everybody okay with that? Super. That's all number 1. Number 2, Paleozoic. Well, if you don't remember, let me get you over to the page in which you may have written this down. Paleozoic and the words that we're going over here, that's on page 151. Nope, page 150. Page 150. Maybe you wrote it down the day we were working on this. Page 150. What does Paleozoic mean?
Ancient life.
Ancient life. Mesozoic, middle. Cenozoic, I'm sorry? Recent life, thank you. Who told me that? Alaska, very good. Page 150 is where you can find these. Page 150. Could you glance at page 150 and tell me how long the Paleozoic lasted? Did you guys write that down? 291 million years. Pretty long. Alexander, what about the Mesozoic? 185.5 million years. And I got the Cenozoic because that's current, 65.5 million years. Whew, these are long time periods. Okay, the fossils. They're in those little purplish boxes back there. I guess they're maroon colored boxes back there. But where else can you find the fossils? Blackboard. Very good. I've got pictures of the fossils on the blackboard. What do I have to know about the fossils? Well, here it is. Common name and era. Now, I have a sheet on that. Let's make sure everybody has that. It looks like this. It says biology fossils. Do you have that sheet? And then check out my Blackboard site. I've got a PowerPoint there that has all the pictures. Most of you have probably seen it already. Okay, moving on. Homologous. Now, guys, does homologous mean, and if you don't know where this is at, it's page 154 at the top of the page. Does homologous mean similar structure, or does homologous mean similar function? Similar function? Structure. And everybody looks, page 154 at the top of the page. Homologous means similar structure. Similar structure. How about analogous? That's similar function. Very good. Okay, here's the last part of this question. Does homologous or analogous represent a closer evolutionary relationship? Which one means you're more closely related, if you have homology or if you have analogy? Homology, very good, homology. Guys, we worked on homology on page 155. All of those vertebrate forelimbs on page 155 are homologous because they have similar structure. Similar structure. Some of the animals are using them for walking. Some of them are using them for flying. We're using ours for object manipulation. Okay, so these forelimbs are being used in different ways, so they're not analogous, but they have similar structure, so they are homologous. Every vertebrate forelimb on page 155 is homologous. Okay, next page, page 156. What accounts for the many differences between chimp and human skeletons? From the neck down, we are similar but different. Do you know why? One reason why we're different than chimps. You know, like we have our head and torso balanced over our pelvis where theirs is thrust forward. I'm just looking at page 157 where we filled out the table. Or our spine is curved, theirs is straight. Our pelvis is short and broad, theirs is long and narrow. All of those, what's the reason why we're different? But why? That's the skull difference. But what about from the neck down? Why are we different than a chimp?
We're adapting to a different way of life.
But what is the way of life we adapted to? That's very good. Walking on 2 legs, they're still walking on 4. So it's our posture. That's the word I would use. Posture is why our spines are different. And our hips are different, and our knees are different, and our feet are different. It's because we're adapted for 2 legs, walking on 2 legs, and they're walking on 4. Everybody okay with that? And I agree we evolved, but I want to know, what's up with that? And it's because we're upright on 2 and they're not. Everybody okay on question number 5? Okay, question number 6. Now, Alexander, help me out with the skulls. And everybody, the skulls are on page 158. Give me 2 reasons why our skull looks different than theirs. Alexander had one of them. What was it, Alexander?
Bigger brain.
Brain size. Very good.
And what we eat.
That's good. They're still mostly herbivores, and we are omnivores. What do herbivores eat? Mostly plants. So they're going to have real big jaws for chewing. We're omnivores. What does that mean?
We eat meat
Meat and plants. So our jaw's not so big. Right here, it's not so big. Okay, on number 6. All right, the skulls. Some of you have been practicing the skulls this morning. That's great. You should be able to name the skulls and then tell me what kind of diet the animal has. So I have this question. What do teeth tell us about an animal's diet? If you see an animal's skull and all of its teeth are sharp, even the ones in the back, what does that mean? Eats what? It's a carnivore. How about if you see an animal skull and all the teeth are ground down so far you can actually see inside the teeth? It's an herbivore. How about if you see something kind of in the middle? It's an omnivore. So make sure you take a look at my skulls and their teeth. Okay, comparative embryology is on page 159. Page 159. It says, how does comparative embryology help to determine relationships between organisms? Okay, so first, if you were looking at 2 embryos and you found that the embryos were very similar, would that mean that they shared close relationship or not close relationship? If their embryos were very similar, would they be closely related, not closely related? They would be closely related because their structure is similar. Okay, what if you compared 2 embryos and they didn't have very many similarities at all? Not closely related. It just makes sense, doesn't it? Or just think how that just makes sense. Okay, good. I like this question very much. Would you expect a human embryo to be more like a chick embryo or more like a pig embryo? Pig. And I agree. But why? Because what are we? Mammals. What are pigs? What are chicks? They're birds. Humans are mammals. Pigs are mammals. Chicks are birds. Here, I'll write that down for you in case you didn't know. Chicks are birds. Yeah, they are birds. So yes, the answer is pig. I like your thinking on that. Very good. What are some basic biochemical molecules that practically all organisms have in common? I'll get us started. I took the easy one. DNA. Give me some more. Something all cells have. If you don't know, feel free to look. Page 160, top of the page. Page 160, top of the page. ATP. Anything else? Enzymes, many enzymes. That's good, like DNA polymerase, RNA polymerase, ATP synthase, some of the enzymes I've mentioned this semester. Anything else? Amino acids, the 20 common amino acids, they're all the same. That's good. You guys know this, you're just not thinking of it. Yeah. mRNA, tRNA, rRNA, all cells have that. You knew that. You just didn't think of it. I know. But okay on this first part of number 8? Okay. How can this be explained? How can you explain that all cells have all of these chemicals and use them in the same way? How can you explain that? How come bacteria have the same kind of DNA I do? Common ancestry. Good. All life has common ancestry. Common ancestry. Okay, the degree of antigen-antibody reaction, page 161. Let me tell you what we did that day. We took a plastic plate, and we put serum in the plates, and then we looked for white stuff to form. Do you guys remember that? Mm-hmm. Okay. And so looking at the plate, and I'm going to do it in a plate, and you have to look at it. You're going to have to tell me who's most closely related to humans, because it's all going to be based on humans, who's most closely related to humans, and who's least closely related to humans. How will you know which one's most closely related to humans? What will you look for? Lots of white. And how will you know who's least closely related to humans? Not much white. Is everybody okay with that? Just want to show you visually what we use so you can remember. Because it's pretty easy. Pretty easy. Okay, on that one. All right, so then the last thing on this one is the cytochrome C clock. Guys, it's on the back of your fossil sheet. Back of your fossil sheet. I especially like question number 3. Is there anybody who doesn't have that? Because I have extra copies up here. Anybody who doesn't have that? Everybody's got this one? Okay. I especially like question number 3. All right, so guys, the next lab, we have just this really big list of definitions. We can get all those definitions from our lecture notes. If you can't find all the definitions in your lecture notes, page 166 is a great page where you can look up some of this. Page 165, whoops. page 166, where you can get a bunch of these words. The reason I drew that line right there is because these things down here are mechanisms of evolution. These are some of the things that cause genetics to change in a population. Mutation, gene flow, which is migration, non-random mating, genetic drift, and natural selection. And your book does an excellent job on page 166 at the top of the page talking about those things. So you can find especially these things right here below the black line on page 166. Okay, number 2. What is the purpose of the Hardy-Weinberg law? Does anybody know why we do Hardy-Weinberg?
Basis comparison of evolution.
Good. That's very good, Alex. Okay, so the idea is that evolution is the change in the genetics of a population, but you can't know if something's changed if you don't know what it was. So it's the baseline for evolutionary comparisons. You got to know that, the purpose of the Hardy-Weinberg. It's baseline for evolutionary comparisons. In the absence of outside evolutionary agents, that means there's no mutation, there's no genetic drift, there's no migration. There's no, everybody's randomly mating. There's no natural selection. So evolution's not occurring. How would you expect gene frequencies of a large population to change over time if there's no evolution? Would they change? Would P's and Q's change if there's no evolution?
No.
No. I believe this is still part of number 2. Is that correct? Okay. Yes. Number 3 is a true or false. The frequency of the recessive phenotype, that means percent little a, in a population equals the frequency of the homozygous recessive genotype. Is that true or false? How often you find the recessive phenotype is equal to little a. Is that true or false? If I say see that you have blue eyes, and I count everybody in this room that has blue eyes, I count you. Does everybody agree blue eyes are recessive? Okay, is that percent little a? Yes, this one's true. That's true. Okay, check out number 4, similar but different. Does anybody see what's different about number 4? Yeah, it's talking about on number 4, it's dominant. All they did was change one word. It says the frequency of the dominant phenotype. How many people are dominant, with brown eyes or green eyes? In a population equals homozygous dominant genotype, percent big A. Is that true? No, because you could have brown eyes but be big A, little a. So this one's false. Okay, so number 3 is true, but number 4 is false. Because everybody realize that if you're looking at the dominant phenotype, you could be big A, but what else could you be? Big A, little a. Everybody okay with that? Good. Okay, I told you this last week. I am not playing around. You really do have to know what every symbol means in Hardy-Weinberg. I'm really not playing around. You really do have to know that P is the frequency of the dominant allele, or Q is the frequency of the recessive allele. We can find these things on page 167. Number 6, given a set of genotypic frequencies from a generation of a population, what would you expect the frequencies to be the next generation if the population is obeying Hardy-Weinberg? Well, first the question is, what does that mean to obey Hardy-Weinberg? That means no evolution. If you're obeying Hardy-Weinberg, that means no evolution. Okay, so I'm saying that if P equals 0.5 and Q equals 0.5, what would it be later on, like 10 generations later, if the population is obeying Hardy-Weinberg? Would be the same. P would still equal 0.5 and Q would still equal 0.5. So if you're obeying Hardy-Weinberg, you're not evolving. Is that okay? So just to make sure everybody got that, if P equals 0.5 and Q equals 0.5 and there's no evolution, what would P and Q be after 5 generations? The same. What about after 10 generations? The same. So if you're obeying Hardy-Weinberg, nothing's changing. Okay, I'm going to do this one in lecture downstairs, so because we are out of time. But you can go ahead and take a look at it if you want to start working on it. That's what I want to put on the board when we go downstairs. 400 fruit flies. 375 have long weeds, 25 have short. We're going to do Hardy-Weinberg on it. Okay, hold on. The difference between the founder effect and bottleneck. Okay, I want to tell you a story. You tell me what I got. Okay, so there's this new island out in the bay. Because, did you know they're dredging the ship channel right now to make it deeper?
Is this part of the story?
It's part of the story. The ship channel is only 45 feet deep, and they're making it 50 because the ships are getting bigger. Okay? So they have to take that dirt somewhere, so they pile it up out there and make islands. So we got a new island out there. Okay, whenever the new island is established, you're going to find some grasses out there. Okay, grasses that weren't there before. What do you call that? Founder effect. Founder effect. Okay, then we have a hard freeze. And many of the grasses die, but some of them live. What do you call that? Bottlenecks. So you're okay on that. Lastly, what are some examples of natural selection? I'll get it started. This is the one I think is a big one. Disease. I think disease kills off a lot of individuals, making the population better adapted to the environment. Jordan? Like the plague. That's a good one, like the plague. Okay, what else? Okay, so defense mechanisms or defense abilities with the spines on the leaves. What else? Weather. Is that a selection pressure? Yes. Too cold, too dry, too wet. Okay, what about predation? Do you think that's very big? Man, being eaten is a huge pressure. So if you can hide from predators or outrun predators, then that would be good for you. That would be a great adaptation. Does everybody see what I'm talking about for natural selection pressures? It's kind of like, what are things that kill off organisms that are not adapted? It's kind of the way to think about it, okay? All right, I'll see you guys downstairs.